Hello everyone, welcome to our channel Medico Mentor. This is part 2 of the video series on the topic Introduction to Immune System. Today I'm going to talk about adaptive immunity. Adaptive immune system mainly consists of lymphocytes, B and T, and their products. This immunity is activated after exposure to an antigen which further improves upon repeated exposure and is specific in action. It is of two types, humoral immunity and cell-mediated immunity. Humoral immunity is mediated by B lymphocytes and their products like immunoglobulins or antibodies. The name humoral is given as the substances involved here are found in the humor, which means body fluids. So it protects against extracellular microbes and their toxins. Whereas in cell-mediated immunity, T lymphocytes are responsible for defense against intracellular microbes and even against cancer. Both B and T cells develop from hematopoietic stem cells in the bone marrow. B cells develop further in the bone marrow, whereas T cells develop further in thymus. Both B and T cells after maturing are still called naive B and T cells. Naive as in they have not encountered antigen yet. After that, they move towards peripheral lymphoid tissue where they get exposed to the antigen and gets activated. Foreign microbes are phagocytosed by macrophages and dendritic cells. We have read about these cells in innate immune system, but they also have a role in acquired immune system. As macrophages and dendritic cells are also known as antigen presenting cells, they have got MHC class 2 receptor. So MHC class 2 presents those part of that antigen to the helper T cells or CD4 positive T cells. Let's talk in brief about development of B cells. It starts from stem cells, then to common lymphoid progenitor cells, then early pro B cells, late pro B cells, large pre B cells, small pre B cells, immature B cells. The point is that in the initial stage, there are no receptors present in the B cells. But later on the development, there starts to develop receptor on the B cells. So IgM is the first antibody receptor to be expressed on B cells, and later on IgD is also expressed. So the structure of these antibodies, as you see in the figure, are quite similar to the antibody structure you have made elsewhere of other antibodies, other classes of antibodies as well. So antibody can exist in two forms. It can be in the serum, as you have seen in humoral immunity, and it can occur as a receptor on B cells. So you have read about other classes of antibodies as well, IgE, IgG, IgA. So how are they expressed on B cells? Because IgM expressing B cells will later on differentiate into plasma cells, and these plasma cells are antibody secreting cells. So they will secrete IgM antibody. So how can you make these B cells to differentiate into plasma cell that secrete other antibodies such as IgG, IgE, and there comes the role of T cells, and the process is known as class switching. Naive B cells, which express both IgM as well as IgD receptors, when it comes in contact with the microbes and again with the help of helper T cells, first it will proliferate. These B cells will proliferate. These B cells can then differentiate into antibody secreting plasma cell. But this plasma cell will secrete IgM antibody since there is no class switching that has occurred previously. Also, it can differentiate into other antibodies such as IgG secreting plasma cells if the process of class switching has occurred. And this class switching process, it occurs by the help of helper T cells. Without helper T cells, there is no class switching. So after class switching, plasma cell will secrete other antibodies such as IgG, IgA, and it will do its function. Similarly, there is another process known as affinity maturation. In this process, those B cells who are expressing high affinity immunoglobulin or high affinity antibody against that antigen will survive, and this occurs through a process known as somatic hypermutation, in which through favorable mutations, though such kind of a receptor antibody are generated which has got higher affinity towards that antigen. So This high affinity antibody expressing B cells will further differentiate into such plasma cell that will secrete high affinity immunoglobulins, and it can be stored as a memory B cells as well. Now moving towards T cell. This is diagram shows CD4 positive T cells as there is CD4 present in the surface receptor. If it was CD8 positive T cells, then there would be CD8 instead of CD4. So CD3 is another receptor, important receptor. It helps to activate T cells. Once antigen binds to T cell receptor, this CD3 is needed to activate this T cells. So similarly, other one is CD28. It binds to other receptor present in antigen presenting cells. So you know that antigen presenting cell through MHC class 2 molecule will present a part of the antigen to T cell receptors, and this CD8 has to bind to other receptor for the signaling to complete. Otherwise signal will be incomplete. So CD28 is very important. Like in B cells, in T cells also it starts developing from stem cells in the bone marrow, then to common progenitor lymphoid progenitor cell, then that common lymphoid progenitor cell moves to thymus for the further development as double negative T cells. These double negative T cells do express T cell receptors, but they are called double negative because they don't express CD4 or CD8 receptors. Once T cell starts expressing both CD8 as well as CD4 receptors, they are called double positive cells. Only those double positive T cells that can react with self-MHC molecule continue to develop, and that is known as positive selection. But those T cells that react too strongly with self-MHC undergo apoptosis. As such, too strongly reacting T cells can result in autoimmunity. So such cells undergo apoptosis, and that is known as negative selection. Then there is downregulation of either CD4 or CD8 molecule, thus resulting in either CD4 positive T cells or CD8 positive T cells, and they move to peripheral circulation. This figure illustrates the interaction between antigen presenting cells and T cells. A part of antigen is presented by MHC class 2 molecule to T cell receptors, but this signal is not sufficient to activate the T cells. In addition to primary signal, there is secondary signal or co-stimulation provided by the molecules such as CD28 on T cells and their ligand B7 on antigen presenting cells. And after that primary and secondary signal, T cells get activated. After primary signal and co-stimulatory signal received with the interaction of antigen presenting cells and CD4 positive T cells, and these cells get activated and change into effector T cells. And examples of effector T cells are Th1 cells, Th2 cells, and Th17 cells. Each effector T cells have their own function, and which effector T cells is going to develop depends on the cytokines released by antigen presenting cells. For example, if the cytokine released by antigen presenting cells is interferon gamma, then the effector T cell that is produced is Th1 cells, and this Th1 cell will further produce interferon gamma, which helps to activate the macrophage, and this activation of macrophage is useful for fighting against intracellular microbes. If the cytokine produced by antigen presenting cells is interleukin 4, then the effector T cells that are produced are Th2 cells, and they produce cytokines such as interleukin 4, 5, and interleukin 13, which helps to class switch and production of antibodies such as IgE and further activation of mast cells and eosinophils, which will help us to fight against parasites. Similarly, if the cytokine produced are transforming growth factor beta, interleukin 6, 1, and 23, the effector T cells that are produced are Th17 cells, which will secrete cytokines such as interleukin 17 and interleukin 22, which helps to recruit neutrophils and monocytes and fight against extracellular bacteria and fungi. This figure summarizes the functions of different adaptive immune cells. B lymphocyte recognizes microbe and it will transform into plasma cells that secretes antibody specific for that microbe, which helps to neutralization of microbe. Antigen presenting cells will present a part of antigen to CD4 positive T cells, and after primary signal and co-stimulatory signal, T lymphocytes get activated and it can change into effector T cells, such as Th1 cells, which will activate the macrophage, Th2 cells and Th17 cells, which will increase the inflammation, and it can also increase the number of B and T cells, which will further boost our immune system. Cells infected by microbes or tumor cells may present a part of antigen to CD8 positive T cells or cytotoxic T cells, and that CD8 positive T cells will secrete porphyrin and granzymes. Porphyrin will make pores in the cells, whereas granzyme will enter into the cell and activate apoptosis and kill the infected or tumor cells. There are regulatory T lymphocytes, which checks the activity of other immune cells and suppresses immune response. At last, let's discuss some of the differences between innate immunity and acquired immunity. So, the components of innate immunity are neutrophils, macrophages, monocytes, dendritic cells, natural killer cells, complement proteins, epithelial barriers, and their secreted enzymes. Whereas the components of adaptive immunity are T cells, B cells, and their circulating antibodies. Innate immunity is present at birth, so the receptors or the cells are germline encoded, whereas in adaptive immunity, the receptors of B and T cells contains V segment, D segment, and J segment, and through recombination of these segments, variety of vast diversity of antigen receptors can be produced. So innate immunity, though is rapid to respond, it is non-specific and there is no memory response. But in adaptive immunity, it takes long period of time to develop. At least it takes 24 to 48 hours, and it is highly specific and it gets refined over time, and there is memory response. Secreted proteins in innate immunity are lysozyme, complement protein, C-reactive protein, defensins, and cytokines, whereas in adaptive immunity, immunoglobulins and different cytokines are secreted. Thank you for listening, and please subscribe to our channel Medicomento for more such educational content. Please share it with your friends and family and help us grow. Thank you.